Hello. So we have talked already about Gregor Mendel and the patterns of inheritance that we see when we look at traits passed from one generation to the next in organisms. And Mendel was able to figure out that those traits are particulate, right? They separate from one another after coming together in one individual. They separate into different gametes, so that's the law of segregation. And he also discovered the law of independent assortment, meaning just because you got one gene from your mother and that goes into one of your sperm, that doesn't mean that genes or factors for other traits are going to go into that same sperm from your mother, right? You may have in that particular gamete the gene for your mom's nose, but the gene for your dad's earlobes, whatever it may be. But Mendel didn't know how that happened. And it turns out it happens because of genes, but genes are on chromosomes. So how do we figure out that chromosomes are the molecules that carry this information, and that the pattern of behavior that chromosomes show during meiosis is what causes this law of independent assortment, segregation, etc.? Well, it turns out that it was because of a scientist named Thomas Hunt Morgan. And Thomas Hunt Morgan is a name that's going to come up quite a bit in biology. He was a brilliant guy. He's actually from Lexington, Kentucky, so not too far away. He did his undergrad at the University of Kentucky and then went on to some other institutions. But he did a lot of work with fruit flies, and he did this in the early 20th century. So at about the year 1900, science rediscovered Mendel's experiments. So Mendel had done these experiments in the mid-19th century, maybe 1860s, but nobody knew about him because he was in a random abbey in the middle of Austria, and his work didn't get publicized very widely. But in about 1900, his work was rediscovered, and when paired with what we knew from Darwin, it went wild, became famous. And so, understanding the patterns of genetic inheritance, we wanted to know, well, why did they behave in that way? And so Thomas Hunt Morgan discovered chromosomes as the carrier of that information. And he used fruit flies for a lot of reasons. One, they produce lots of offspring. They have fast generations every 2 weeks, and they only have 4 pairs of chromosomes. So if he was studying chromosomes and whether they were in fact what carried the information, then it was pretty simple to follow from one generation to the next. So he noted phenotypes as wild type, which is the one that you typically saw in most fruit flies in the wild or in a lab, and he would also note mutant phenotypes. So phenotypes that would pop up occasionally that you didn't see very often. For instance, white eyes. So generally, if you catch a fly, and we've all seen fruit flies, buzzing around our old banana or something, they tend to have these red eyes, but every now and then, at least in a lab setting, you will see mutants that pop up with white eyes. And he decided to breed these red-eyed and white-eyed flies just like Darwin did. He got true breeding red flies and true breeding white flies, and he crossed them. And in the F1 generation, they all had red eyes. And this made a lot of sense based on what he now knew of Mendelian inheritance, right? This meant that red was the dominant allele and white was a recessive allele. And in the F2 generation, he would cross hybrids, so individuals that were clearly heterozygous, that had a red-eyed parent and a white-eyed parent. And as he expected, based on Mendelian inheritance, he got about a 3-to-1 red-eyed to white-eyed ratio. So you did have white eyes that would pop up in this F2 generation. But all the white-eyed individuals were males, right? And Mendel did not predict that. And so Morgan, who had also been interested in the determination of sex, and it had been discovered recently that it had to do with the Y chromosome, figured that the rest of this information must be passed on chromosomally too, specifically eye color in this case, but he expanded that beyond eye color conceptually. Right, so if you look at this, we have red-eyed and white-eyed parents who are homozygous red, homozygous white. Put them together, you get a heterozygote, and they're all red, right? All these individuals are red. But if you cross two of those heterozygote red individuals together, 3 red to 1 white, but all of the whites are males. And so he figured that meant it had to be on a sex chromosome, specifically the X chromosome, because males, if they have one of those white-eyed alleles, recessive white-eyed alleles on an X chromosome, they don't have a second X chromosome where they can mask it, right? Where they can just be a carrier. Right? Males can't be a carrier for recessive disorders that are on X chromosomes. And so you can see the F1 and F2 generation Punnett squares down here. I'd encourage you to take a look at those. And so any trait that is on one of the sex chromosomes, so X or Y, rather than chromosomes 1 through 22, we call sex-linked because the patterns of inheritance you see are different, or at least they're more specific based on sex. So you can imagine in humans we have X chromosomes and Y chromosomes. That's how sex is determined. And the X chromosome is relatively large, Y chromosome is pretty small. The Y chromosome contains a gene that causes your developing gonads to become testes and to make testosterone, and that codes for sort of the male characteristics that we think of. Females don't have one of those Y chromosomes. If a trait is coded for on the Y chromosome, then clearly only males will get it, right? That said, very few traits are on the Y chromosome because it's tiny, right? It really has that SRY gene, which codes for maleness, and not much else. That said, the X chromosome is a pretty big chromosome, and it codes for a lot of things besides sex. And therefore, for traits that are X-linked, so that's sex-linked chromosomes specifically on the X chromosome, X-linked traits, you'd think maybe females would get them more often, but they don't. Because again, females, if it's a recessive disorder, have a second X chromosome. And on that second X chromosome, they might have a dominant gene that will mask that recessive allele. Whereas males, if they get an X chromosome with a recessive trait, they don't have another X with another version of that gene to mask it. So this is a look at the X and Y chromosome and their difference in size. And the difference in size is indicative of how many functional genes there are in there. By the way, I posted a video I posted several videos for you, but one of them was about sex determination. We are chromosomal, right? X, Y, not all organisms are. Right? Some organisms are, and they use different chromosomes. Like birds use different chromosomes. Some organisms use temperature. Some organisms use population density. That impacts which sex the offspring will become. Bees use a haplodiploid system, where any diploid offspring is female and any haploid offspring is male. Right? They literally have a different number of chromosomes between males and females, and that becomes very interesting when you get into social behavior and the evolution of that social behavior. We'll talk about more in Bio 132. So again, a gene that's located on either sex chromosome is a sex-linked gene. If it's located on the Y, we call it Y-linked, although that is not common. But lots of genes are located on the X chromosome, and we call those X-linked. In both cases, males are more likely to show a recessive trait that is sex-linked. X-linked genes, again, more common in males because females need 2 copies. A male only needs 1 copy because he does not have a second X chromosome that would have a second version of that gene that might be dominant and might mask it. These are some very general Punnett squares that don't show a specific trait but show you what a sex-linked trait, or specifically an X-linked trait, would look like if you were to put it in a Punnett square. And I encourage you guys to do some practice with these. I posted some practice on Blackboard and had you do some of those for your most recent assignment. Some examples of recessive X-linked disorders are color blindness. So color blindness, there are different kinds, but most of them are X-linked. And chances are decent that you guys know somebody who's colorblind, and chances are also pretty good that that person is a male. Now, females can become colorblind, but they can only become colorblind if their dad is colorblind, because they have to have gotten, the mom has to at least be a carrier, right, and have a colorblind recessive gene on one of their X chromosomes. But they also have to get a colorblind X chromosome from dad, and that means that dad must be colorblind. Duchenne muscular dystrophy is X-linked recessive. Hemophilia is X-linked recessive. So you might think, logically, all right, well, males only have one X chromosome, females have two. How does that work, right? Do males not make enough of the products of the X chromosome? And it turns out, no, males and females generally make about the same amount of product, because males and females both only have one functional X chromosome. Females will inactivate one of their X chromosomes. But which one is different depending on which cell you're in. And so in every cell, they have two X chromosomes. One of the two of them will become non-functional. It will condense into what's called a Barr body, which is an inactive version. And but again, it's random, right? Which X chromosome becomes inactive is random for each different cell. And so you can get, for instance, in some cats, you have a gene that codes for fur color, and in each hair cell and in each little patch, you can have different X chromosomes that become inactivated. And if you're heterozygous, that means you have patches of different color and hairs that are different color on your fur. So this is an example of how you get a calico cat. In chapter 14, and also at the beginning of this chapter, we talked a little bit about the law of independent assortment, which means that just because you got one particular trait from your mom, maybe her nose, doesn't mean you will also get your ears from your mom, right? You may get your mom's nose and your dad's ears, right? And we know that that means that just because chromosome 7 from the maternal chromosome 7 goes left during meiosis doesn't mean the chromosome 8 will go left. The chromosome 8 from mom might go right? And so every gamete is filled with some mix of maternal and paternal chromosomes, and we get independent assortment of traits. But do we really get independent assortment of traits? And the answer is not always. Some genes tend to move together. And it was also Thomas Hunt Morgan that found this. Again, brilliant scientist. He found that some traits are linked, and he specifically found body color and wing size. So he crossed a fly that was wild type for both body and wings, so normal long wings and a gray body. And he found flies that were mutant, so had the recessive traits for body color, meaning black, and wing size, meaning vestigial wings, so little wings that weren't functional. And he crossed them, and in the F1, right, he got all, individuals that were dominant, and then he did an F1 cross, and he found something interesting, right? Theoretically, it shouldn't matter to your wing size what body color you got, but it did, right? So let me move myself up here. Turned out that he got, most of the individuals, if you were gray, were likely to have long wings, right? And if you were black, right, were more likely to have short wings.